Kicking off the list at number 10, EnviroPig. The EnviroPig was a transgenic pig developed in Canada at the University of Guelph, home of the griffins and the Frankenswine. Awesome, gotta love it. Welcome to Guelph. Scientists from South China's Agricultural University originally wrote a paper about the transgenic pig that can grow faster than normal and, on top of that, have less of an impact on the environment. Lovely, live fast, little piggies. Now, normally these hogs would release a high percentage of nitrogen and phosphorus because they haven't evolved to digest those nutrients in their meals. These nutrients end up in their waste, which subsequently ends up in our lakes and rivers, and then in our mouths and eyes and nose. It would pollute the water with phosphorus, and that's when the EnviroPig scuffles in to save the day. Scientists alter their DNA in order to produce more phytase, in order for Frankenswine to fully digest all the nutrients, instead of leaving us the crappy ones, pun intended.
In our number 9 spot today, we have web-spinning goats. While many of us have dreamed of becoming Spider-Man, it turns out the only ones who get that luxury are goats. Researchers from the University of Wyoming have developed a way to incorporate a spider's silk spinning genes into goat's milk. This would then allow scientists to be able to harvest the silk proteins from the milk in order to use them. You might be wondering why, and that's a super valid question because it seems pretty random, but as it turns out, it actually might have some pretty exceptional uses. Spider silk is a super valuable naturally occurring material because it not only is strong, but it is flexible as well. These types of proteins can be used for medical things like making artificial ligaments and tendons or eye sutures, but it can also be used in other ways like to help build parachute cords or bulletproof vests somehow. Why didn't we just get that silk from the OG source? Well, you need a ton of spiders, and when you put a bunch of them together, they usually just kill each other, so naturally, goats were the next option?
Are you tired of losing your pet rabbits every time there's a blackout? Well, fear no more, this horrific treatment may just solve that problem. Back in 2000, an artist by the name of Eduardo Cac created this glow-in-the-dark rabbit in collaboration with French geneticist Louis-Marie Hodenbein. It was referred to as the GFP bunny, and it was this cute little albino floof that would glow under any blue light. Now, this glowing gal is named Alba, and what Hodenbein did was take the GFP gene found in a jellyfish, a certain type called Aquaria victoria, which rhymes, which is always awesome, but it would glow green when exposed to blue light. Now, Alba was a one-of-a-kind experiment, and it used the word "was" with sorrow. Alba sadly passed away in 2002, but her death has brought up a few questions. A US reporter called experts in France, where Hodenbein worked, and they were told that Alba had passed, but they didn't provide any substantial evidence onto how. No reason was given as to why this rabbit passed away. Now, you would think the world's first glowing rabbit would deserve more than, I don't know, maybe a voicemail to say, hey, it's dead, see ya. Maybe she's still alive. Maybe Hydra has her in some basement. Only time will tell. We'll see.
In our number 7 spot today, we have banana vaccines. Remember as a kid when you were sick and had to get medicine and it sucked, but then you found out it was the banana flavored kind and suddenly things weren't so bad? Well, this is absolutely nothing like that. Scientists have been able to successfully engineer different foods like bananas, lettuce, potatoes, and carrots to produce different vaccines, but apparently bananas are the most ideal candidate for this sort of thing. What's crazy is that with bananas, when an altered form of the virus is injected into the banana sapling, it ends up acting almost exactly like a traditional vaccine. Not the mRNA one that we're becoming familiar with the pandemic, but the kind used for things like hepatitis and cholera. This is because once the banana sapling is injected, the virus's genetic material then becomes a permanent part of the plant's cells. From here, as the bananas grow, the cells of it produce the virus proteins, but not the infectious part of the virus that makes you sick. So your body can now learn these proteins and then build up the antibodies needed in order to fight it. Honestly, this one seems a little weird on the surface, but I am all for the idea of vaccines one day not being given through needles. I don't like bananas either though, so guess I'm shit out of luck.
Number 6, spicy tomatoes. Picture this, you're at a barbecue, you're eating burgers with your peers, you're having a great time, and then somebody next to you starts coughing and you ask if they're okay, and they're like, oh, I'm fine, it's just, this tomato's just oh so spicy. Huh? Yeah, spicy tomatoes, they're a thing, let's talk about it. Scientists in Brazil and Ireland have cracked the condiment code, or so they thought. Using CRISPR, they created the first ever tomato that is naturally spicy. The trick is in its DNA. Tomatoes already carry the appropriate genes to produce capsaicin, which is the same ingredient that makes your mouth burn every time you eat a hot chili pepper. Some are on board with the spicy tomato, dare I say they're even excited to use this for some Saturday spicy salsa. Honestly, I'm all set for this. I ate a junior chicken and I'm like, oh, can I get a glass of milk? I'm not good with spice at all. It's all fun and games until a Greek salad lights up your lips.
In our number 5 spot today, we have DIY crisp CRISPR-Cas9 kits. If you haven't heard of the controversial Josiah Zayner, he is the eccentric biochemist who is the owner of a startup company called the Odin. Josiah is no stranger to the world of genetic engineering, and in fact, he has done some experiments on himself when he injected himself with modified cells to become more muscular. He did this by removing the gene for myostatin, and without this gene, muscles keep growing. Regardless of this interesting experiment, what I really want to talk about is how the Odin has released CRISPR-Cas9 kits that allow you to experiment with genetic modifications yourself. I'm sure this is probably harmless, but it just seems like this kind of power should stay in the hands of those who know exactly what they're doing. Maybe that's just the skeptic in me, but there's something about a DIY genetic modification kit made by a guy who tried to modify his own muscles that just seems a little weird. No judgment, just not my personal cup of tea.
Number 4. Salmon. This photo shows a genetically engineered salmon next to farmed Atlantic salmon. They're both the same age, they're both 18 months old, but the one in the back looks like it's been bench pressing for 9 months. It's clearly much larger, and that's because AquaBounty's genetically modified salmon grows at double the speed. No more fake IDs for those salmon, they're getting in quick. Canadian scientists took the gene that regulates these growth hormones from salmon, combined it with an ocean pout's DNA, and now we have the ability to simply grow these fish up a few years. Now the taste is the same. In fact, apparently it's even better. This modification gives the Atlantic salmon a fresh taste all year round. The same company that I mentioned earlier, AquaBounty, also plans on using these fish to mass produce eggs at its research hatchery in PEI. They then want to ship these eggs to Panama, where they'll grow again twice as fast, but this time in a land-based facility, where they'll wait approval by the FDA. The thing is, neither Canada or the US has done nearly enough safety trials. It's just AquaBounty, so we really don't know any long-term effects that this will have on us or the ecosystem. We can't have 9-foot salmon swimming up our rivers every day. It's a bit alarming, I think.
In our number 3 spot today, we have Dolly the sheep. You may have heard of Dolly the sheep before, and that is because she made history in 1996 when she was born. This is because she was the very first mammal to be cloned from an adult somatic cell. The process of cloning to create her involved nuclear transfer from a cell taken from the mammary gland. Basically, they transferred the nucleus from the adult cell into an unfertilized premature egg that had its nucleus removed, and boom, cloned sheep. This was huge for the scientific community because of the fact that it not only showed that cloning can be successful in mammals, but also because it showed that a clone organism can be produced from a mature cell from a specific body part. While Dolly made history, she also sadly only lived a relatively short life. She ended up being euthanized just before her 7th birthday because of the fact that she had a progressive lung disease as well as arthritis, both of which are said to not be linked to her cloning origins. I guess it's kind of cool that Dolly existed, and the fact that we have these scientific abilities is amazing, but there's also something pretty ominous about it. I just hope we use these powers to do something like bringing back extinct animals rather than just some weird mad scientist type stuff.
And in at number 2, venomous cabbage. As if cabbage wasn't already not appealing, we've now thrown venom into the mix. Lovely. Venomous cabbage, why do we need this? Well, it starts with scorpion venom. It's one of their most vital tools when it comes to self-defense, obviously, so taking that gene and combining it with cabbage could limit our use of pesticides, but at the same time, still prevent those fat caterpillars from harming our crops. Hey, if you have a caterpillar problem, just poison them. There you go, guys. Sounds appetizing too. The cabbage would then naturally produce this scorpion poison, so if any pest came along looking to have a work lunch, they would be long gone before they could even leave a Yelp review. Poor little dudes.
In our number 1 spot today, we have CRISPR babies. This is probably the most well-known case of human genetic engineering, and probably not for the right reasons. On November 25th, 2018, He Jiankui, who is a biophysics researcher, announced, here on YouTube, that his team had successfully created the world's first genome-edited babies. These babies, Lulu and Nana, were born from genetically modified embryos that had been made to be resistant to HIV. Here's the thing, no one is arguing about whether or not this could potentially be an amazing thing for people. The argument is about whether or not it is ethically correct to genetically modify a human before they're even able to be a human yet, especially when the potential risks and long-term effects of this kind were completely unknown. This study was also conducted without the public discussion of the scientific community, which again is the source of quite a bit of controversy. In the end, he ended up being sentenced to jail time as well as being forced to pay a fine. 2 others he worked alongside were also found guilty for having, quote, forged ethical review documents and misled doctors into unknowingly implanting gene-edited embryos into 2 women. At the end of the day, scientific advancements are very cool and exciting, but not at the cost of doing the right thing the right way.
For number 10 now, we have the Vicente mouse. There's no easy way to say this. The Vicente mouse was a mouse with a human ear on its back. Well, not technically. The ear mouse was created by Charles Vicente and his colleagues at the University of Massachusetts in 1997. The ear on its back was actually an ear-shaped piece of cartilage grown by seeding cow cartilage cells into a biodegradable ear-shaped mold that was then implanted underneath the mouse's skin. But yeah, it's an ear, basically. So what was the point of all of this? Well, when the picture started circulating around the internet in the late 1990s, many people used it in protest against genetic engineering. However, no genetic manipulation was actually performed here. The whole point was to show that transplantation of fabricated cartilage into humans is possible. We have the Vicente mouse to thank for that, even if it does look very weird. Next up at number 9 now, we have the see-through frog. I love this one. In 2007, Japanese researchers succeeded in producing see-through frogs. The reason so that they didn't have to kill and dissect them anymore in order to study their organs, blood vessels, and eggs. How amazing is that? In modern times, dissections have become increasingly controversial. Many animal rights activists have called for more humane methods of studying frogs without having to perform dissections. Lead researcher Masayuki Sumida said, "You can watch organs of the same frog over its entire life as you don't have to dissect it." The researcher can also observe how toxins affect bones, livers, and other organs at lower costs. These frogs were created from rare mutants of the natural Japanese brown frog. They bred pale frogs together until the offspring were completely translucent. At the time, the researchers also said they could inject the frogs with an illuminating protein that would cause parts of the frog to glow when cancer starts. Imagine if they did that with humans. Interesting, but perhaps a little bit creepy. Moving on to number 8 now, we have Dolly the sheep. Some of you may have heard of this one before. Dolly was a sheep. She was also the first mammal to be cloned from an adult cell. She was born in Scotland after scientists from the University of Edinburgh did years of research and failed attempts. She was created using the technique of somatic cell nuclear transfer, where the cell's nucleus from an adult cell is transferred into an unfertilized egg cell that's had its nucleus removed. The cell is then stimulated to develop by an electric shock. The cell was then implanted into a surrogate mother and was eventually born into a healthy, normal-looking. The cell was then implanted into a surrogate mother and eventually became a healthy, normal-looking sheep called Dolly. It's a risky procedure and often results in stillborns or calves that last just a few days or weeks. However, Dolly was stable. The scientists waited almost 7 months to announce her to the world, but when they did, the news was everywhere. Many people called it the breakthrough of the year. This cloning paved the way for other large animal clones such as pigs, deer, horses, and bulls. In January 2018, scientists even successfully cloned macaque monkeys, the first primates ever cloned. Is human cloning around the corner? Next up at number 7 now, we have the glowfish. These are fish that glow in the dark. They aren't locked away in some lab away from the public eye. You can buy them. They are on the market. They are 100% for sale. In 1999, scientists in Singapore took the glowing gene of a jellyfish and inserted it into the embryo of an Indian zebrafish. The result was glowfish. They glow in both natural and ultraviolet light. This wasn't the ultimate goal for the scientists. They actually wanted to develop a fish that could detect water pollution by glowing in the presence of environmental toxins. They were then approached by Yorktown Technologies, who bought the right to sell the fish worldwide. They are sold in bright red, green, orange, yellow, blue, and purple fluorescent colors. Their names include Electric Green, Moonrise Pink, Cosmic Blue, and Galactic Purple. The FDA approved them for sale in the US once they assessed that they were not going to end up in the food chain. But still, if you see one, maybe don't eat it. That's just my advice for today. Next up at number 6 now, we have Ruppy. In 2009, scientists at Seoul National University created the world's first transgenic dog. Her name was Ruppy, and she glowed under ultraviolet light. Of course, the gene to glow in UV doesn't naturally occur in dogs. They got the gene from sea anemones. Ruby was created by cloning fibroblast cells that express a red fluorescent gene produced by the sea anemones. It was hoped that transgenic dogs like Ruby would become effective stand-ins for the study of human diseases. Professor Lee Byung-chun led the research team and said, "What's significant in this work is not the dogs expressing red colors, but that we planted genes into them." Of course, he can say that, but the media and the world became obsessed with the idea of a dog that glowed when you shone a UV light on it. Other scientists criticized the process, saying transgenesis is laborious, expensive, and slow. Moving on to number 5 now, we have the sudden death mosquitoes. That's the name that's been given to these these mosquitoes because essentially scientists have engineered them to possibly bring an end to their own species. Sounds crazy? This is how it works. They genetically modified male mosquitoes with lethal genes. Once released, the males mate with the females in the wild and release these lethal genes which kills them or kills the offspring before they can develop into adulthood. If successful, techniques like this could wipe out huge swathes of mosquito populations around the world or even bring them to extinction. The result isn't just because mosquitoes are annoying, it's to stop them from spreading the likes of dengue fever, which has no known cure and has put nearly half of the world's population at risk of infection. Of course, this is good news for humans. 2.3 million infections and 25,000 deaths could be prevented every single year. However, as bad as they are, some people are questioning whether humans have the right to intentionally kill off mosquitoes, an entire species. What do you guys think? Coming at number 4 now, we have the fast-growing salmon. In 2015, America's FDA approved Aqua Advantage salmon for human consumption. These are Atlantic salmon with an added gene which allows them to grow all year round instead of only during spring and summer. The intention has been to increase the speed at which the fish grows without affecting its ultimate size or other qualities. These fish can now reach their market size in 16 to 18 months instead of the usual 3 years. Despite the approval by the FDA and other governments around the world, it's unlikely that you'll be finding these special salmon everywhere. Back when they were approved, there was just 2 land-based facilities that were allowed to grow the fish. One day though, they could become the norm. Next up at number 3 now, we have the EnviroPig, a name that I absolutely love. In 2010, researchers at the University of Guelph created genetically engineered pigs dubbed the EnviroPig. Now on the surface, they look just like normal pigs, but it's not about the outside, it's about what's going on inside them. You see, normal pigs produce a lot of phosphorus in their urine and feces. That then finds its way into lakes, rivers, and oceans, causing algae blooms. The outburst of algae rapidly depletes the water's oxygen levels, creating vast dead zones that kill fish and other aquatic life. Not good. Pigs are often blamed for contributing to exactly this. These specially engineered EnviroPigs produce up to 65% less phosphorus in their urine and feces. This would make things better for the environment, but also for farmers. You see, many farmers had to abide by strict laws that allow no farms to release nitrogen or phosphorus runoffs. They usually have to store all the waste and wait for it to be treated and recycled as fertilizer. This is all just added costs for farmers. It's hoped that the EnviroPigs could change all that and do some good for the environment at the same time. Moving on to number 2 now, we have the sterile pink bollworm. Pink bollworms are a nightmare for cotton farmers. Every year they destroy cotton crops around the world. It got so bad that the US Department of Agriculture began tests to see if they could stop the worms in their tracks. They began growing a lot of pink bollworms in laboratories. That might sound counterproductive if your aim is to reduce their numbers, but wait, the male bollworms had actually been genetically engineered to be totally sterile, meaning even if they mated with a female, no offspring would come about. The worms were then released back into the natural population where they competed with wild male bollworms for the females. Scientists hope that if enough of these sterile males mate with the females, the number of offspring would be greatly reduced, providing some relief for cotton farmers around the world. And finally, at number 1 now, we have the see-through goldfish. In 2009, Japanese researchers succeeded in creating a goldfish that was so translucent you can see its heart beating inside its body. The aim was to create a fish that could be studied without the need for a dissection, the same reason for the frogs that we talked about earlier. Yutaka Tamaru, a professor of the study, said, "You don't have to cut it open. You can see a tiny brain above the goldfish's black eyes." Scientists created the fish using selective breeding. They took 2 natural fish who had slightly paler skin than the rest and bred them together. They continued like this with the offspring until the skin became so pale it was see-through. The fish are expected to live up to 20 years. They can grow as long as 10 inches and weigh more than 6 pounds. That's much bigger than fish used in similar experiments. They hope that for the people studying these fish, they can now see their whole life cycle take place underneath their see-through skin without ever having to hurt them. Right, starting off at number 10 now, we have the twin studies. Joseph Mengele was the chief medical officer at Auschwitz concentration camp during the Holocaust. Far from being a doctor to the people there, he used them as part of his sort of twisted medical studies. Mengele had an obsession with twins. From 1943 to 1944, he performed experiments on almost 1,500 sets of imprisoned twins. Just 200 of them survived. One study involved injecting different dyes into the eyes of twins to see whether it would change their eye color. Another involved sewing twins together in some sort of sick attempt to create conjoined twins. Some of the other studies are far too graphic to even talk about in this video. Next up at number 9 now, we have the poison labs. This is the name given to the Soviet Union's secret testing labs for poisons. During the 1950s, the researchers there would test deadly poisons on prisoners who came from the gulags. That translates to enemies of the people. These poisons included mustard gas, ricin, digitoxin, curare, cyanide, and many others. All of them are deadly to humans and many of them are very painful. The goal of these studies was to find a tasteless, odorless chemical that could not be detected after death. In a final sick twist to this story, the prisoners were told that the poison was actually medicine for them. Insane. Next up at number 8 now, we have the Stanford prison experiment. In 1971, psychologist Philip Zimbardo led a research team to study human behavior when it came to authority and inmates in a prison setting. He set up a mock prison and got 24 volunteers to take part in it. They were assigned as either prisoners or the guards watching over them. Everything was designed to look and feel like a real prison, from the uniforms to the cells and even the ID numbers. The guards started to get way more involved in their roles than the researchers thought they would, verbally and then physically abusing their prisoners. By just the second day, the prisoners had already started to rebel. The guards put it down with brutal violence against them. The prisoners began to get emotionally traumatized, and on the 6th day, the experiment was ended 8 days early. Moving on to number 7 now, we have the Aversion Project. This experiment was a medical torture program in South Africa conducted by Dr. Aubrey Levin during the 1970s and 1980s. It was the military's attempt to root out homosexuality from the army, as it was seen back then as a mental illness. Over the years, 900 gay servicemen and some women were sent to the infamous Ward 22 for sexual reassignment surgeries, some of them as young as 16 years old. These surgeries involved chemical castration and electric shock treatment. The surgery attempted to change the men into physical females. Apart from these surgeries being horrifically unethical, they were also botched quite often, leaving the victims cast out into the world without the hormones needed to maintain their new identities. Coming at number 6 now, we have Project 4.1. This was a US medical study of people living on the Marshall Islands who were exposed to radiation from a nearby nuclear bomb test in 1954. The study effectively saw them as guinea pigs in a radiation experiment. Many people accused the US government of having their primary goal as studying the people and not actually helping them. The poor residents of the Marshall Islands suffered miscarriages, stillbirths, developmental difficulties, and impaired growth. The government knew the American public would not react kindly to news of this experiment and ordered everyone involved to keep it a secret. But as you can tell, secrets always get out. Next up at number 5 now, we have the Monster Study. With a name like that, you know it's not going to be good. This study took place at the University of Iowa in 1939 by Wendell Johnson. They took 22 orphan children and split them into 2 separate groups. One group was given positive speech therapy. That means they were praised and encouraged as they became more fluent in English. The other half got the exact opposite. They were belittled, even bullied every single time they made a mistake. This had a serious negative effect on the children. Some of them even stopped speaking altogether. Their mental health and academic studies suffered greatly. The university tried to sweep all of this under the rug, but eventually the details came out. They were criticized heavily for damaging vulnerable orphan children, maybe permanently, just to prove a theory. The university issued a public apology in 2001. At the number 4 spot now, we have the Willowbrook State School. This was a school in New York for people with mental disabilities. Starting from the late 1950s, medical researchers noticed there were large hepatitis outbreaks there. They studied it to gain a better understanding of the illness. However, the researchers seemed to cross a line when they began feeding live hepatitis viruses to 60 healthy children in order to study them. They watched their skin and eyes turn yellow, their livers got bigger, and they began to vomit and even refuse to eat. The researchers justified all of this by saying, well, they were going to get hepatitis anyway, but history has definitely condemned them. Moving on to number 3 now, we have the Tuskegee experiment. This clinical study by the US Public Health Service took place for an astounding 40 years, beginning in 1932. It involved offering 399 poor black males who had syphilis a treatment. However, the researchers withheld the treatment as their real aim was to just study the progress of syphilis on humans. In 1947, penicillin became available as a treatment. Some of the victims began to look elsewhere for help, but the researchers told them not to and continued to lie about their true condition. When the study finally ended in 1972, just 74 of the 399 test subjects were still alive. 28 of them had died of syphilis, 100 from other complications. 40 of their wives had also been infected, and 19 of their children were born with congenital syphilis. As part of a class action lawsuit, the US government paid $9 million to the surviving participants and their descendants. For many people though, the pain can never be healed with money. Next up at number 2 now, we have mustard gas. In 1943, the US Navy exposed its own sailors to mustard gas in order to test the effectiveness of their new protective clothing for it. They wanted to see how it stood up in a real life situation and not just in a lab. Perhaps the most infamous of these experiments involved 17 and 18-year-old males still in boot camp who were asked if they wanted to take part in an experiment that would shorten the war. Of course, many of them said yes, but it wasn't until they reached a lab that they were told it would involve mustard gas. Some of them left at this point, but many of them didn't. Most of the participants suffered extreme external and internal burns. They were largely ignored by the Navy after that because they didn't want to draw attention to this awful experiment. And finally, at number 1 now, we have Unit 731. This was the secret chemical warfare research unit of Japan during World War II. They wanted to study the human body in perverse ways, and mainly did so on Allied prisoners of war as well as Chinese civilians. Experiments included limb amputation to study the effects of blood loss, and sometimes they would even try and reattach amputated limbs to the other side of the body just to see what would happen. One other famous experiment there involved freezing all of the subject's limbs and then amputating them one by one until only the victim's head and torso remained. This is only really scratching the surface into the horrors that Unit 731 brought upon its many victims.
Kicking off the list at number 10, the aviator suit. When it comes to new ideas and new inventions, it almost seems messy at first, all the time. It's always a horrible start. And today's list is quite messy, a lot of horrible experiments done with groups of unaware subjects. To kick this grim one off, we'll look back to the early 1900s. We'll start with Franz Reichelt, a known inventor of the time. He invented his own version of a wearable aviator suit. It was a parachute. It was made of 320 square feet of fabric. It wasn't light at all. And in 1912, Franz wanted to test this new invention out in public, but instead of using a dummy and throwing that off the Eiffel Tower, Franz wanted to be brave and he strapped himself in. And the rest is history. Dark, horrible, fast history. Number 9. The Hoffling Hospital Experiment. This experiment happened all the way back in 1966, so a bit more recent, in a time where the rules of psychological experiments, well, didn't really exist. Really. I was gonna say they were kind of loose, but no, they were not really a thing at all. Because of this, the nurses that were all part of this experiment had no idea that they were participants, which nowadays is totally illegal, very illegal. The night nurse would receive a phone call during the shift, and on the other end would be Dr. Smith, who's actually the researcher in this scenario. He would ask the nurses to check the medical cabinet to see if they had a drug called Astrotin, which was actually a drug that was made up for this experiment, and it was actually just a placebo, so don't worry too much. The Astrogen would clearly state the maximum dosage of 10 milligrams, but Dr. Smith would ask the nurses to administer 20 milligrams. They were told that the doctor was in a hurry and he would sign the papers later and all that jazz. This just had to get done that night. This had to be delivered to the patient ASAP. If the nurse decided to give the patient the drug, they would be breaking 3 rules. They're not allowed to accept instructions over the phone, the dose was double the maximum limit stated on the box, and the medicine itself was unauthorized. Big 2. And it was not on the ward stock list, so it shouldn't be used in the hospital in the first place. Big 3 red flags right there. Out of the 22 unknowing nurse participants, 21 of them went to administer this. Yeah, big oof on that one. Yikes. To be fair, that's the entire point of this test. This is nothing towards the nurses. I'm not ripping on them at all. I mean, this was 1966. We didn't know quite a bit back in the day, you know what I mean? This really showed the pressure nurses are put under randomly in the middle of the night, if anything. Number 8, UCLA schizophrenia experiment. Starting in 1983, UCLA researchers Michael Gitlin and Keith Neutraline went to great and pretty unethical lengths just to see how people who are suffering from schizophrenia relapse. Yeah, basically they're trying to figure out if there is a way to predict the relapse, or a psychosis episode of some sorts. Now, sounds like a good idea on paper, I guess, but the way you go about things, historically, come on, we could have done a lot better. Unfortunately, this experiment involved recruiting hundreds of participants who were all being treated for schizophrenia at the time, and then just taking them off their meds completely. Yeah, as I said that, you at home probably went, ooh, yeah, that's exactly right. What a horrible idea. Like, we're going the opposite way here. We're not supposed to do this, right? There was no suitable plan and order for when they could return to their medication. And they also didn't do a good job making sure these patients were actually safe during these experiments. They're like, yeah, let's just take people off their meds, see what happens. It's horrible, it's one of the worst. Unfortunately, the results were as bad as you thought and proved to be fatal when one of the participants, Antonio Lamadrid, ended up taking his own life. Again, not sure what we expected here in this sense. An article from 1994 also said the doctors failed to get proper consent from the patients as well. Just a cherry on top of this horrible experiment. Just when you thought it couldn't get worse. There you go. Number 7. Psychic driving. British psychiatrist Donald Ewan Cameron is to blame for this one. Remember that name. Don't listen to anything he says. He created the psychic driving concept that the CIA found interesting, to say the least. Basically, psychic driving was a procedure that subjects patients to a continuous, repeated audio loop of something that's intended to change their behavior. Just the same line or the same whatever over and over thousands of times. And through the course of their treatment, they would also be paralyzed while being exposed to the loop message. Yeah, the worst thing ever, right? So the CIA heard about this and started sending money to fund Cameron's experiments, but he actually didn't know it was coming from the CIA because they used a fake name. Classic CIA. Cameron would subject patients to paralytics and electroconvulsive therapy, stronger than usual as well, just to make things worse, just at a horrible limit. The experiments were mostly conducted on patients who entered the institute for more common problems like anxiety disorders or postpartum depression, and ended up leaving with permanent effects from his actions. These included things like amnesia, being unable to speak. Some people forgot their parents' names and thought that the interrogators were actually their real parents. Just horrible things. Their minds were just so confused. Brutal stuff. Number 6, Go pills. Here in Toronto, we have Go trains, Go buses, but not Go pills. Not yet. Hopefully not ever, actually. Yeah, move over 5-hour energy. We got some competition on the horizon. Staying awake is challenging, even when you're doing something you absolutely enjoy, like watching a movie. You'll still fall asleep. It just happens, we're human. My dad fell asleep in a theater once in the moving chairs. I couldn't believe it. Guy's 3D glasses were falling off of his face. I was in shock. But he works a lot. He works crazy hours, he's older. That's where Go pills were supposed to come in handy. Workers who have these long shifts and work late hours to pay the bills. This pill was supposed to keep you up for 40 hours straight, so a bit much, I'd say. The US Defense Advanced Research Project Agency, or DARPA, which is much less air coming out of my mouth, is currently funding this pill. But what if it's mistreated? I can't even begin to imagine the negative effects something like this could have on people. We could barely handle Tide Pods as a community. You know what I mean? It's kind of a bad idea, I vote. Number 5. The Milgram experiment. When learning about our past in further detail, like the Trinity test for example, and how that led to Hiroshima and Nagasaki, it's important to understand why these things happened and why these people did this, or more so how people are influenced into doing these horrible things historically, especially in large numbers. So the Milgram experiment was put in place to get an idea. How far will people go when it comes to obeying instructions, specifically if it involves a third party being hurt? Well, Stanley Milgram, Yale psychologist, created this test. There's 3 important roles for this one. You need the teacher, the experimenter, and the learner. The learner is disguised as the main test subject, but really, they're actually in on the whole thing. The real subject is over here. The teacher, the one administering pain to the test subjects. So they're told by this higher figure, whoever, to keep upping the pain every time they get a question wrong. They're like the rule maker. So the real test here is to see how far humans will go when hurting others under direct authority. It's important to note that the subject also isn't actually getting shocked at all. Again, they're in on it. They play it up. They play it up more and more just to see what's going on. These tests were underway in 1961 in a basement at Yale University. In college, I did improv. That's wild. That's a wild after-school curricular activity. Number 4, Unit 731. The Imperial Japanese Army's Unit 731 conducted some pretty horrible experiments during World War II that certainly are going to shock anyone who learns about them. This is wild. This definitely belongs on this list. The experiments were meant to be done as a way to prepare for biological warfare, but the process was horrible and extremely inhumane, as are most of these things. Different medical schools and universities all provided doctors and other research staff to help conduct these experiments. Big group effort, nice, way to go guys. They used both prisoners and civilians as test subjects. There were a bunch of different experiments that were conducted during this time, some of which involved injecting them with pathogens like cholera or anthrax, or operations with just no anesthesia, just weapon testing, all painful stuff, really the worst of the worst. Number 3, MK Ultra. We have to include a mind control project. Do you believe in mind control? Some claim they're born with it, like an X-Men, so if that's you, you know where to meet. MK Ultra, where do I start? Okay, it was originally a secret CIA project that lasted from 1953 to 1973, where they ran hundreds of experiments to US citizens. They gave them hallucinogens in an attempt to crack mind control, or sorry, as they called it, information gathering. Yeah, it's like what Apple says. Oh, we're just gathering information. Yeah, okay. In the 50s and 60s, around the Cold War, the United States believed that the Soviets, Chinese, and or North Korean agents were all using mind control in the war. That was their best explanation towards these brainwashed prisoners of war in Korea at the time. Yeah, it's gotta be brainwashing, nothing else, right? Horrible tests were being conducted in universities, hospitals, and again, in prisons. The happenings of these projects weren't fully known to the public until years after it ended. It was a huge, horrible secret. The agency destroyed most MK documents back in 1973 when it officially ended, so we think we know, when in reality we probably know little to nothing what actually occurred. Number 2. The Stanford Prison Experiment. Perhaps the most famous psychological experiment gone wrong. The Stanford Prison Experiment. Let's talk about it. It started August 14th, 1971. It was led by university psychology professor Philip Zimbardo. Again, another university study. My gosh. I mean, to be fair, Hogwarts was also messed up, but Hogwarts wasn't real now, was it? The Stanford Prison Experiment was very real. The experiment took volunteers and divided them into 2 groups. One group of prisoners and one group of guards. And they placed all these volunteers into a fake prison that was created for the experiment, okay? The experiment aimed to see if and how quickly humans would turn evil, under the right conditions of course, with the right amount of power. Yeah, pretty twisted, right? It's basically a group version of the Milgram experiment. It was a test to try and answer the question of if humans are inherently good or inherently evil. And after just 6 days, we found out. Yeah, 6 days later, the experiment needed to be concluded because the guards began absolutely tormenting the prisoners in the worst ways possible. It really showed the kinds of things humans can be capable of, even after just a short amount of time, less than a week. And humans are like, oh, I'll be evil. And finally, coming in number 1, facial expressions experiment. Yep, a wild one to close her off. The facial expressions experiment. Okay, back in 1924, a psychologist with, you guessed it, the University of Minnesota, classic university stuff, he wanted to conduct an experiment to study facial expressions, right? Sounds pretty harmless so far, dare I say sounds a little silly. More specifically, he wanted to see if everyone's expressions of emotions were all the exact same. Does happiness look the same on everybody? Does sadness? What about fear or shock, disgust, anger? What about those? So he recruited some volunteers from campus and then painted the lines of their facial muscles black. And then he exposed each participant to different stimuli in order to photograph their reactions. He wanted to compare all these results side by side. The stimuli at hand was intense. This guy wanted big reactions. He included showing them adult films, he exposed them to ammonia, he made them touch reptiles, and of course horrible things that I can't talk about here on the tube of you. You know what I mean? Horrible, horrible stuff. All in the name of facial expressions. What an awful experiment. What a dark one.
Starting us off at number 10, we have the quagga. Once a prolific subspecies of zebra across South Africa, the quagga was sadly hunted to extinction in the 19th century. The decline in population all started after European colonists took to South Africa and hunted the species nearly into the ground as it competed with domesticated animals for forage. By 1878, the wild population was decimated, although some were taken to zoos in Europe in an effort to rehabilitate, but tragically in the end, the breeding programs were unsuccessful and the last captive quagga died in Amsterdam in 1883. That was until about 100 years later when the quagga became the first extinct animal whose DNA was analyzed, and it was then that scientists discovered just how close a match they were to the plains zebra. By 1987, the Quagga Project began, and their mission is to recreate the phenotype of hair coat pattern by selectively breeding the genetically closest subspecies, which, as we now know, is the zebra. The first foal of this project was born in 1988, and since its early days, they have seen quite a bit of success. If all goes well, the selectively bred quagga-like zebras will be released back into the wild. Coming in at number 9, the Cuban macaw. Prior to the 15th century, these vibrant and colorful birds could be found all over the main island of Cuba. But when Europeans landed, they had other plans for these parrots. Although indigenous populations had hunted the species prior to the colonists' invasion, once the Europeans arrived, the amount soared. Many were hunted or traded, while others were sent back off to Europe to live as cage birds, and eventually, by the late 19th century, between the rampant overhunting and habitat destruction, the very last of the Cuban macaws died, making the species extinct. extinct. But according to the British writer Errol Fuller, aviculturists are rumored to have bred birds similar in appearance to the Cuban macaw using the genes of a sister species. So who knows, we could be seeing them fly around Cuba in the near future. Coming in at number 8, the Arabian oryx. Historically speaking, the Arabian oryx could have been seen roaming around throughout most of the Middle East, but by the time the late 19th century rolled around, you were not likely to see one outside of Saudi Arabia, and in the 1930s, the only remaining populations were found either in the Negev Desert in the north or the Rub' al Khali in the south. Tragically, it only got worse from there. Starting in the 1930s, it became fairly customary for Arabian princes and oil companies to hunt down the animal, and eventually the hunts grew to employ as many as 300 vehicles at once. By the middle of the 20th century, the northern population was effectively extinct, and finally the last Arabian oryx died in 1972. Thankfully, a few people had the forethought to send a couple off into captivity, and they were effectively reintroduced into the wild a few years later. That being said, they are still an endangered animal, but interestingly, it is the first animal to revert to a vulnerable species after being declared extinct. So it's definitely getting better. Coming in at number 7, Caspian tigers. During their prime, Caspian tigers could be found in Turkey and through much of Central Asia, including Iran, Iraq, and northwestern China. But with the Russian colonization of Turkestan during the late 19th century, their population began to be threatened. The first problem was obviously that the tigers were being hunted by large parties of sportsmen or military personnel. Until the early 20th century, the army was used to clear predators from forests around settlements and potential agricultural lands. In fact, until World War I, about 50 tigers were killed in the forests each year. By the 50s, they became an officially protected species, but even so, by the 1970s, the last remaining Caspian tigers were gone. However, according to recent findings that the Siberian tiger is the closest relative to the Caspian tiger, discussions have been started about introducing the Siberian tiger into a safe space in Central Asia. And hopefully, if they get it right, the tiger would adapt and live successfully where the Caspian tiger once roamed. Coming in at number 6, heath hens. Once an extremely common bird, heath hens were a subspecies of prairie chicken that could have been found just about anywhere across North America in colonial times. However, like many other species, the population was deeply affected by colonizers who hunted them extensively for food. In fact, it has been speculated that at the first Thanksgiving, it was actually heath hens that were served, not wild turkey. However, by the time the 18th century rolled around, the heath hens developed a reputation as being the poor man's food, as it was cheap and plentiful. And as the hunting continued to soar, the heath hens became more and more of a distant memory. By the 1870s, the animal was virtually extinct from the mainland, with only a few hundred left on Martha's Vineyard, until eventually the late 19th century put a hunting ban in to try and keep the species alive after an estimated 70 remained. This worked for a while, but due to a myriad of problems, the last remaining heath hen died in 1932. But there is still hope for them to return. As they are closely related to the prairie chicken, scientists have started researching projects aiming at the de-extinction of the animal using DNA from preserved cells as a basis for restructuring the DNA of greater prairie chickens. So with any luck, heath hens could be roaming around soon again. Coming in at number 5, oryx. Considered to be the wild ancestor of modern domestic cattle, oryx were a species of large cattle that once lived in Europe, Asia, and North Africa. And when I say large, I mean large. Apparently, oryx could reach a height of 6.6 feet at the shoulder and weigh up to 3,000 pounds. However, due to hunting and habitation loss, the oryx became extinct when the last individual died in 1627. 1987. However, recreating the extinct species may not be an out-of-this-world thought according to scientists. Starting back in the 1920s, Heinz Heck, a German biologist, initiated a selective breeding program where he attempted to breed back the oryx using several cattle breeds. Essentially, the idea was to breed out what they had been domesticated for, and the result was called Heck cattle. By the 1980s, herds of Heck cattle were released into the Netherlands, and since, Heck cattle have been crossbred with other European cattle breeds in the hopes of creating a more oryx-like cow. Coming in at number 4, the dodo. Maybe the most recognizable of any extinct species due to just how straight up bananas this bird looked was the dodo. Once upon a time it was found exclusively on the island of Mauritius, but what was most fascinating about the dodo was that it evolved without any natural predators. However, like many animals that evolved in isolation, the dodo was entirely fearless of humans. However, this fearlessness, coupled with its inability to fly, unfortunately made the dodo an all too easy target for sailors. Although contrary to popular belief, it wasn't just hunting that took out the dodos, but the introduction of other animals like monkeys and pigs into their habitat, who destroyed their nests and forced them into a competition for limited resources as well. So, have these cartoon-looking birds actually been brought back since their 17th century extinction? To be honest, not technically. But scientists are trying. Beth Shapiro, a professor of ecology and evolutionary biology at the University of California, said that she has completed fully sequencing the dodo's genome from an ancient DNA based on genetic material extracted from dodo remains, and the next steps are in the works. Next up at number 3, the Tasmanian tiger. The Tasmanian tiger was once a carnivorous marsupial native to the Australian mainland as well as the islands of Tasmania and New Guinea. But beginning in the 19th century, they were perceived as a threat to the livestock of farmers, so overhunting was eventually the largest factor in their demise. Sadly, the last of the Tasmanian tigers was illegally captured in 1936 by Elias Churchill and sold off to a zoo where she stayed until her death. But a return of the Tasmanian tiger could very well happen in our lifetime. In 1999, the Australian Museum began a cloning project for the animal, an endeavor that was initially dismissed as a publicity stunt. But in 2002, researchers successfully extracted replicable DNA from the specimens. Now, fast forward to August of last year, and the University of Melbourne announced it will be partnering with a company called Colossal Biosciences to attempt to recreate the animal using its closest living relative and return it to Tasmania. Coming in at number 2, the woolly mammoth. I'm sure we're all familiar with the gigantic fuzzy ancestor to the elephant because, well, it's probably one of the most famous extinct species recorded. The last isolated population of woolly mammoths lived on Wrangel Island in the Arctic Ocean until roughly 4,000 years ago, and famously the first actual remains of the behemoth were found in Siberia in the late 18th century. Interestingly, at first, the researchers studying them were incredibly confused because they could not understand how a warm climate animal such as an elephant could have wound up in such a freezing cold area. But eventually they put all the pieces together and came to the realization that what they had found were the remains of an extinct animal. Amazingly, since those first few skeptical years, thanks to the cold temperatures in the Arctic, the carcasses of the extinct creature have been super well preserved by ice, allowing scientists to access the DNA in ways that is not always possible. And thanks to that, it is apparently in the works to try and bring the beasts back into the world. In fact, after a genome project for the mammoth was completed in 2015, it has been proposed the species could be revived through various means. So while it hasn't happened just yet, by the sounds of it, it could be any day now. And last up in our number 1 spot, the Pyrenean ibex. While there are many genetic testings in the works along with breeding of similarly extinct species with the hopes of adaptation, if we are talking about an actually extinct animal that has been successfully brought back to life, then the Pyrenean ibex is in a league of its own. During its prime, it was often found in areas like France, Portugal, Spain, and Andorra. However, starting in the late 19th century, their population began to dwindle until eventually the Pyrenean ibex ibex was officially declared extinct in 2000. But the amazing thing about this animal is that unlike any of the others on this list, 3 years after the last one died, scientists used its frozen cells to clone a calf, making it the first and only animal to have a living specimen exist post-extinction. Now, the caveat is that sadly the clone did not survive long after birth. It actually died a few minutes later due to a lung defect. But it did prove that it could be possible to actually bring a species back from the dead.
Starting off in our number 10 spot, we have Dolly the sheep. We had to start this list off with Dolly today because she really set the stage for everything on this list to come. You may have heard of Dolly the sheep before, and that is because she made history in 1996 when she was born. This is because she was the very first mammal to be cloned from an adult somatic cell. The process of cloning to create her involved nuclear transfer from a cell taken from the mammary gland. Basically, they transferred the nucleus from the adult cell into an unfertilized premature egg that had its nucleus removed, and boom, cloned sheep. This was huge for the scientific community because of the fact that it not only showed that cloning can be successful in mammals, but also because it showed that a cloned organism can be produced from a mature cell from a specific body part. While Dolly made history, she also sadly only lived a relatively short life. She ended up being euthanized just before her 7th birthday because of the fact that she had a progressive lung disease as well as arthritis, both of which are said not to be linked to her cloning origins. I guess it's kind of cool that Dolly existed, and the fact that we have these scientific abilities is amazing, but there's also something pretty ominous about it. I just hope we use these powers to do things like bringing back extinct animals, maybe, rather than some weird, mad scientist type of stuff. In our number 9 spot today, we have Samrupa. After the birth of Dolly the sheep, scientists everywhere began trying their hand at cloning, and that is when Samrupa was born. Back in 2009, Samrupa was born in India and was a Murrah buffalo, which is known as being the best known milking buffalo, since these animals can be capable of producing 35 kilograms of milk a day. The birth of Samrupa marked a breakthrough in the country because it put India on the map as one of the few countries at the time who had the capabilities to clone, and this was all done using a new at the time landmark technique which was called hand-guided cloning. The scientists who helped to create Samrupa worked on the calf for 4 years, so it's clear that they were all very excited about its birth. Unfortunately, however, shortly after Samrupa's birth, things went seriously wrong. Just 5 days after birth, Samrupa unfortunately succumbed to a lung infection and passed away. While the scientists weren't able to save the animal, it is very clear that its birth and short life helped scientists in remarkable ways that went on to inform them in future experiments and projects. In our number 8 spot today, we have the Pyrenean ibex. The Pyrenean ibex is an animal that went extinct around 2000. In a horrible turn of events, the last one was a female named Celia, and she was killed in an awful falling tree incident. A subspecies of the Spanish ibex, the Pyrenean ibex were native to the Pyrenees Mountains on the border of Spain and France. Back in the medieval ages, their population was reduced drastically to an endangered level. This is due to 2 things: being hunted as well as the spread of human disease. Flash forward to 2003, however, and scientists tried to bring them back to life. This is the first extinct creatures that scientists ever tried to clone. That is absolutely crazy. For 7 minutes. DNA from Celia, the last living individual of the species, was taken and implanted into the womb of a domestic goat. From here, the clone was in fact born, but due to lung complications was unable to survive for longer than 7 minutes. It was a short life, but a monumental one that definitely broke new ground in the scientific world. In our number 7 spot today, we have Garima 1. Garima is similar to Samrupa that we already talked about, and in fact she was born in the same place, the National Dairy Research Institute, just shortly after Samrupa, and luckily her birth did end up being more successful considering she made it past the first year of life. She was actually cloned on June 6th, 2009, and things seemed to be going quite well until around June and July of 2011. Garima was created through the same hand-guided cloning technique as Samrupa, so it's clear that the technique was evolving and getting better, but things still unfortunately went awry. When things took a turn for the worst, doctors struggled to figure out exactly what was the problem, but eventually it was revealed that her problem was with her heart. Researchers revealed that her heart was abnormally overweight and much larger in size than other buffaloes her same age, and this likely led to the complications she faced that sadly culminated in her passing in August of 2011. In our number 6 spot today, we have the Argali. In the earlier days of cloning experiments, scientists attempted to clone an Argali, which is also known as a type of mountain sheep. These sheep are known to roam the highlands of western East Asia, the Himalayas, Tibet, and the Altai Mountains. The reason they wanted to clone these sheep is because they are important to the ecosystems they live in, and unfortunately they are a threatened species. Now, I know there's controversy over the cloning of threatened or extinct species, but personal opinions aside, they were making this attempt to see if this could be a viable way to potentially save a species. Unfortunately, however, these attempts would prove to be futile. Using the same techniques used in the instance of Dolly the sheep, the somatic cell nuclear transfer, while they believed it would be promising, all attempts to clone the mammal failed to produce any viable results, and thus modern science wasn't able to assist in the continuation of this species. But in good news, while the species is still threatened, it is still around and not quite endangered, although its life is mainly threatened due to trophy hunters and ranchers. In our number 5 spot today, we have the gar. A gar is a species of wild cattle that is native to South Asia and Southeast Asia, and unfortunately they are listed as a vulnerable species on the IUCN Red List, and they have been since 1986. This is exactly why, in 2001, scientists decided it was time to try and clone one. With a population that has declined over 70% in the last 3 generations, they seemed like a perfect candidate to become the first ever endangered species to be cloned. In 2001, at the Trans-Ova-Genetics Center in Sioux Center, Iowa, they were able to birth a clone gar. They obviously used gar DNA, but used a domestic cow as a surrogate mother to carry the clone. This of course was a huge breakthrough, but unfortunately the life of the gar was cut short as the calf passed away within 48 hours of its birth. Although this is obviously terrible, I guess the upside is that it is believed that the calf passed due to dysentery, which is not thought to have been related to the cloning process, which does mean that although the outcome wasn't as predicted or hoped, the cloning could still be considered a success. In our number 4 spot today, we have Injaz. With a name that means achievement, she clearly was exactly that, because Injaz was the first camel clone ever produced. She was born back in 2009 at the Camel Reproduction Center in Dubai, and she certainly was a proud accomplishment for all of those who worked to create and raise her. In fact, the cloning of Injaz was such a success that she lived for over a decade. Her birth prompted the center to continue cloning camels, which they did about 20 to 25 times a year in the time after Injaz was born. She broke ground once again when she proved that cloned camels could in fact conceive and give birth naturally, and she did so a few times, but in the end it would be this that led to her demise. On her 4th calf, as she carried it, it grew to be too big for her to carry, which unfortunately led to dystocia late in her pregnancy. Scientists did their best to save her, but unfortunately they lost both of them. Everyone who worked to care for Injaz was rightfully extremely upset at her passing, but they clearly used her and her success as a starting point for what was to come. Now the center is turning out dozens of cloned camels every year, some going for $55,000. Not sure how I feel about that, but you know, whatever. In our number 3 spot today, we have the Bantang. Back in 2003, scientists at the Advanced Cell Technology of Worcester, Massachusetts were ecstatic when they successfully birthed a clone Bantang, which is an endangered species of cattle. To do this, they actually took frozen tissues from a male of this species that passed away many years earlier in 1980. They used these skin cells and inserted the nuclei into cow eggs, and a cow was used as a surrogate mother. They attempted this with 16 pregnancies, 2 of which came to term. With the success of one clone did come the birth of another identical twin who was born to a different cow mother, but despite being carried to term and birth, this clone was not as expected. It was twice the normal birth weight, which quickly proved to be a very negative sign. As for the clone who was considered a success, it was hand-raised at the San Diego Wild Animal Park's Infant Isolation Unit. Unfortunately, however, due to an injury that the animal got when it was less than 7 years old, it did end up passing away, which meant that sadly the clone only lasted for about half the expected lifespan for for this species of animal. In our number 2 spot today, we have the woolly mammoth. We would certainly be missing a very important animal on this list if we didn't include the woolly mammoth. These guys were of course a species of mammoth, and in fact they were one of the last in line of the mammoth species. Because of the fact that in both Alaska and Siberia there were frozen carcasses discovered, they're actually one of the most well-studied extinct creatures. Exactly why or how they were driven to extinction is widely debated, and another thing people like to debate is whether or not we should try and bring them back. There was a genome project completed in 2015, and since then it has been proposed the species could be revived through a few different means, but no one has yet taken the leap. Here's the thing though, there's a new company that is pretty set on de-extincting woolly mammoth, and they have a new investor. This investor is called In-Q-Tel, and they are registered as a non-profit venture capital firm that is funded by the CIA. They claim that the interest in this company is less about mammoths and more about the capability to do such crazy projects. I know this is more like a future project that might happen, but when we're talking about a huge animal like the woolly mammoth, we have to bring it up. Okay, I live in Canada. That's where the woolly mammoth would also live, I feel like. I don't know, I feel like it just has consequences for me, so I'm concerned. In our number 1 spot today, we have the Morian viviparous tree snail. This was a species of air-breathing tropical land snails that were endemic to French Polynesia, and their near extinction was caused by a chain of events that happened after something humans did. The African land snail was introduced into Tahiti in 1967 as a food source, but it quickly escaped and began to destroy crops. Biologists wanted to attempt to control the African land snail, so they decided to introduce the rosy wolf snail to the area in 1977. This went absolutely haywire, as the rosy wolf snail didn't just control the population of African land snails, but rather started to eradicate all of the snails that were native to the area, which of course includes our little friend. So this one little introduction led to them being totally extinct in the wild. These snails still exist in captivity, and there have been attempts to re-release them into the wild, maybe clone them to boost their population numbers, but unfortunately the rosy wolf snail continues to prey upon them, so at this point researchers are unsure if it will ever be possible for them to live in the wild again.
Starting off at number 10 now, we have the Manhattan Project. Some of you guys may recognize the name of this one. It was the secret name for America's project to produce the atomic bombs that brought World War II to an end. Harry Daghlian was a young physicist who worked on the project. During an experiment on August 21st, 1945, he was attempting to build a neutron reflector manually by stacking a set of 4.4 kilogram tungsten carbide bricks in an incremental fashion around a plutonium core. Unfortunately, he dropped a brick which set off a chain reaction. He tried to stop the reaction, but by the time it was over, he received a high dose of neutron radiation. Harry received intense medical care for severe radiation poisoning. His mother and sister even flew out to New Mexico to care for him, but Harry fell into a coma. He died 25 days after the accident. His death resulted in new safety regulations when handling radioactive material. Next up at number 9 now, we have the dioxin tests. In 1965, dermatologist Dr. Albert Kligman conducted a study on behalf of the US Army and some pharmaceutical companies. They wanted to know how human skin reacts to harsh chemicals, a process known as hardening. Most of the information about these tests has since been destroyed or just covered up. We do know that he described his patients as acres of skin to experiment on. We also know he used dioxin on them, the active ingredient in Agent Orange. This happened around the time the US was covering Vietnam with Agent Orange in an attempt to push the fight against the Viet Cong in their favor. To speed up the experiments, Dr. Kligman injected his victims with a reported 468 times the recommended safe dose of dioxin. After that, not much more has been uncovered. It's thought there are still about 70 of the patients out there still. Their identities and current condition are known. Next up at number 8 now, we have Operation Sea Spray. In September 1950, the US Navy spent 6 days spreading the bacterium Surretia marcescens into the air about 2 miles off the coast of California. That bacterium lives in soil and water and is best known for its ability to produce a bright red pigment. This trait makes it useful in experiments because it's so bright, it's very easy to see where it is. The project was called Operation Sea Spray. Its aim was to determine the susceptibility of a big city like San Francisco to a bioweapon attack by terrorists. In the following days, the military took samples of 43 sites to track the bacteria's spread. They found that it had infested the whole city city and all the surrounding suburbs as well. During a test, residents of San Francisco inhaled millions of bacterial spores. The military deemed the experiment a success. They got to see how far chemical warfare could spread, and anyway, they knew the bacterium wasn't harmful, or so they thought. People started getting urinary tract infections, and it was even linked to the death of a patient recovering from prostate surgery. This experiment wasn't even public knowledge until 1976, a full 26 years after the event. Moving on to number 7 now, we have Project MK Ultra. This is a particularly famous one that you guys may have heard me talk about in a previous video. During the 1950s and 60s, at the height of the Cold War, the US feared that communist countries were using mind control techniques to brainwash US prisoners of war in Korea. They wanted in on the action. So in response, the CIA authorized Project MK Ultra in 1953. The secret operation was aimed at developing a defense against drugs or other manipulators that could control human behavior. Such was the secrecy of this project that even today, not much is known about it. But we do know this: the project involved more than 150 human experiments involving psychedelic drugs, paralytics, and electroshock therapy. Sometimes the test subjects were aware they were participating in a study, but shockingly, many did not, even when the hallucinogenic effects started kicking in. Can you even imagine how scary that would be? Many of the tests took place at universities, hospitals, or prisons in the US and Canada between 1953 and 1964. The CIA didn't keep many records though, and any good ones were destroyed when the program came to an official end in 1973. Moving on to number 6 now, we have North Korea. Some of the things we've all heard about North Korea may not be true, but some of them may be even worse than we could ever imagine. In 2014, a former North Korean officer stepped forward to say that the nation's military were using mentally and physically disabled children as test subjects in chemical experiments. He said this was the last straw that caused him to defect and leave the country. The man was called Im Cheon-young, and he said he was taken to special training at the military academy. It was there where they taught him how to confuse the enemy without revealing your own forces, how to carry out assassinations, and how to use chemical weapons. He was close to defecting on this alone, and then came the field learning. This involved testing biological and chemical weapons on so-called objects. At first, these objects were mice, but soon they were using humans. Im said that he saw with his very own eyes disabled people, sometimes children, being killed by these chemical weapons just to show the soldiers how they worked. One expert said this is not surprising. Anyone who visits the capital will never see disabled people because they are allegedly taken away as children and incarcerated in special camps. Perhaps these evil experiments are what the camps are all leading up to. Moving on to number 5 now, we have Project QKHILLTOP. This was a CIA project developed to study Chinese brainwashing techniques, which they used to develop new methods of interrogation. The research was led by a man named Dr. Howard Wolf. Now, he was a researcher at Cornell University's medical school. Dr. Wolf requested the CIA provide him with information on imprisonment, deprivation, humiliation, torture, brainwashing, hypnosis, and more. Based on this, the team began to formulate a plan. They wanted to develop secret drugs and various brain-damaging procedures. According to a letter that he wrote, in order to fully test the effects of this, Wolf wanted the CIA to make available suitable subjects. Once they had provided these poor souls to test on, Wolf also detailed the next step his research team would take. They would then assemble, collate, analyze, and assimilate the information, and will then undertake experimental investigations designed to develop new techniques of offensive, defensive intelligence using a potentially useful secret drug. Basically, the whole thing is just the stuff of nightmares. I have no idea how this got through. Nothing more is really known about the test subjects, or victims as they are often referred to now. Moving up to number 4 now, we have Dr. Loretta Bender. This doctor has gone down in history for her experiments on children. She was a child psychiatrist who specialized in children believed to have schizophrenia. For almost 3 decades, from 1942 to 1969, she was a highly respected expert in her field. Her preferred choice of treatment was electroshock therapy. These days, the practice has been banned in most of the world as it's seen as ineffective, cruel, and borderline torture in some cases. Still, this was a different time, and that's what Dr. Bender did to the children. Her methods involved interviewing and analyzing a sensitive child in front of a large group of people. She would then apply a gentle amount of pressure to the child's head. The theory was any child who moved with the pressure was showing early signs of schizophrenia. These days, that's referred to as total pseudoscience, but it took a while to figure that out. By the time her treatments were shut down, Bender had used electroshock therapy on over 100 children, the youngest of whom was just age 3. Reports described her as uncaring and the study as unethical. The shocks can cause memory loss, nausea, headaches, jaw pain, or even serious heart trouble, even in kids. Some say all of this was a result of Bender's misunderstood childhood that she had, and she was the product of her own environment and family. Now that is proper psychology, I'm just saying. Moving on to number 3 now, we have the Burke and Hare murders. William Burke is a guy who has probably tainted my surname forever. Along with William Hare, these two men from Northern Ireland were famous grave robbers in Scotland. One day, an old man they knew died, and to cover his outstanding debt to them, they decided to sell his body for medical science. Edinburgh University gave them £7 and 10 shillings, a handsome sum in those days. With that, they were hooked. When another associate they knew fell ill, they couldn't even wait to see if he would die, and so they suffocated him in his bed. Again, they sold his body to the university for money. The killing spree began. The pair murdered and sold bodies almost as their full-time jobs. They killed an old grandmother with an overdose of painkillers. Hare even killed his own blind grandson by breaking his back across his knee. Eventually though, their whole operation was discovered when they got too sloppy. William Burke was hanged in front of a cheering crowd of over 25,000 people. Fittingly, after being put on public display, his body was donated to medical science. Next up at number 2 now, we have J. Marion Sims. This doctor was once praised as the father of modern gynecology. Now his reputation has been tarnished forever. It was found that he practiced these surgical techniques that made him famous on enslaved women. They included Lucy, Anarcha, and Betsy. The rest of them are unknown. He performed 30 surgeries on Anarcha alone, all without anesthesia. His legacy has long been questioned for the disturbing ethical questions that it raises, especially by those who believe that he used black women as medical guinea pigs without their consent. The Black Youth Project 100, a group of activists aged between 18 and 35, staged protests at a statue of Sims in New York. They wore hospital gowns splashed with red paint dripping down their legs. In a Facebook post, they explained that Sims had purchased black women slaves and used them as guinea pigs for his untested surgical experiments. He repeatedly performed genital surgery on black women without anesthesia because, according to him, black women don't feel pain. Despite his very inhumane tests on black women, Sims was named the father of modern gynecology, and his statue currently stands right outside the New York Academy of Medicine. And finally, number 1 now, we have the pregnant women. In January 2018, it emerged that after the Second World War, the US military gave nearly 1,000 pregnant women radioactive iron by their doctors without their knowledge. This was all part of a series of experiments to test the effects. Between 1945 and 1947, researchers at Vanderbilt University conducted this sick experiment that was funded by the US Public Health Service. All of the women were poor and had no knowledge of the experiment whatsoever. Afterwards, they were not even informed that they had been part of the study either. The radioactive iron was given as part of a medical cocktail by health officials that they trusted. Their aim was to record the absorption of iron during pregnancy. However, in the years since then, investigators have said this too was a lie, and that the real reason was for the military to learn more about radiation exposure. In the 1990s, Vanderbilt University finally acknowledged and investigated the experiments. The story broke when 2 women and one of their daughters filed a lawsuit against the university for exposing them to radiation. One of the women was Emma Kraft. She said at a Senate hearing that the experiments caused the death of her 11-year-old daughter due to cancer. The true effects of this study are still being investigated today.